One was being involved in the Human Genome Project and seeing the potential from the industrialization of sequencing. There was really amazing gains in productivity that came from industrializing sequencing, and then really surprising changes in the way we carried out biology by having the capacity there. And so I saw a real opportunity to apply the same principles to another part of the biological process, which is acquiring and manipulating DNA molecules. And the other part of the experience that drove me in this direction was being involved in a biotechnology company. Okay. For about 5 years, and seeing the amount of effort that went into acquiring and manipulating DNA molecules just for the very conventional parts. Yeah. No, yeah. Okay, let me concentrate. Okay.
I'll worry about this.
So this will be, we've got another minute or two. So today, a lot of what we do is focused on substituting for activities that could be carried out in other ways with conventional molecular biology. You know, we've done thousands of genes, but most of those are for people who are doing much more conventional biology than people are talking about here. But I think that our guess is that 10 years from now, most of what people are doing will be much more the kinds of things that the speakers here have been talking about. So I apologize for...
It's not your fault.
Okay. Yeah. I was just on vacation in Mexico. About Blue Heron already. So the company was started in 1999, and we launched a commercial product in 2001. And right now, as I said, a lot of our customers are in the conventional biotech and pharmaceutical companies, and we have about 70% of those at one time or another have bought genes from us, and we've produced thousands of genes in the last 3 years. And I just wanted to give you a little bit of background on gene synthesis. It's something that many of you are familiar with, but some of you may not be. It's been in use since the late 70s, but it's not something that's been widely used until fairly recently, and it really has to do with a couple of challenges that have made it difficult to do, and there's two real errors in the process that make it a little bit more difficult to do than it might seem right off the bat. And one is the errors that arise from the chemical synthesis of oligonucleotides. So oligonucleotide chemistry is an extremely efficient process as organic chemistry goes, but there's an error rate of about 1 in 300 in the base changes, either deletions or substitutions. And the other sort of complication in assembling genes is that ligation and hybridization don't proceed with perfect fidelity, so there's not a perfect Watson and Crick interaction in every case, and so it's possible to get gross errors of order. And both of these kinds of problems impact the reliability and increase the cost of the process. And there have been really three general approaches to doing gene synthesis. There's a sort of one-pot Pcr approach or ligation approach, what we call convergent assembly, and a solid phase assembly. And all of these have been described in the literature, I believe. So the one-pot or Pcr ligation approach is basically to synthesize oligonucleotides that span the gene and then combine multiple oligonucleotides into a single reaction and create the fragment or the part of the fragment that you desire. And it has a number of compelling advantages. It's simple to do and it works often. And it's the technology that nearly all of the commercial providers and most of the people in academic labs use. And several groups, including George Church's group here, are integrating PCR assembly with oligosynthesis on chips. And that's very appealing because it could lead to a very low-cost assembly technology. there are a couple of limitations from the point of view, at least of commercial applications, and one is that some of the sequences are difficult or impossible to create that way, things that don't PCR, for instance. And then there's been, at least in the past, problems with getting high-quality oligos off of chips. So convergent assembly is a somewhat different approach. You start out with oligos, again, that span the gene you want, but it involves a series of ligation and purification steps, which each involve only 2 fragments. So you take 40-mer duplexes, combine those to make 80-mers, purify those, combine 80-mers to make 160-mers, and so on to build a final fragment. So convergent assembly is, it involves a series of simple, very reliable steps, and it'll work on almost any sequence. So we found very few sequences that cannot be built this way, but it is slower and more expensive than PCR. So the technology that we're using for gene assembly now, we call solid phase gene assembly, and basically it takes from the logic of oligonucleotide assembly, oligonucleotide synthesis. So we add oligos sequentially to a solid support with intervening wash steps. So in this example, you're adding the first duplex, you attach that duplex to the solid support, wash away the side reactions and the unincorporated oligos, add the second duplex, attach, and so on to assemble the full-length fragment. So that has a number of advantages. It has the same sort of simplicity that the convergent assembly has. You can drive the reactions with a molar excess, which isn't possible in any of the homogeneous solutions. You can wash away side reactions at each stage. And it's readily automated. And one other advantage is that I think it's very amenable to scaling down the size and reagent volume, and this may lead to a very low-cost approach for oligosynthesis. I'm sorry, we're getting some kind of a, that's what it is. So we've developed a solid phase robot for doing gene assembly that was supported by an SBIR grant from the NIH. And really it puts a room full of activity into a single box. We use it to assemble fragments up to 750 base pairs in length, and then use a recursive strategy after that of assembling those cloned fragments. So assembly of whole genes involves putting oligos together, as I described, to build what we call subtargets. Those are cloned, sequence verified, and then released. And then those subtargets are assembled with a similar process to build the final clone. And if you compare the gene synthesis methods, PCR assembly will work on many or most sequences. The costs are low and the speed is relatively high, so you can complete the process in weeks. Convergent assembly will work on essentially all sequences. We believe that we can build almost any sequence that way. There are sequences that are difficult to clone in E. coli, things with long repeats or toxicity. And solid phase assembly, again, is extremely reliable. Each step in the process is very simple, but because it's automated, the costs are low and the time is faster than PCR. So our technology, the technology we bring to bear on gene synthesis, is a combination of things. There's algorithms, the software designs for breaking a gene sequence down into a series of oligonucleotides that can be built back together. The convergent assembly technology, the solid phase assembly technology, and with convergent we built thousands of genes and hundreds with the solid phase, so we're convinced that both of them are highly reliable. And we also use a variety of error correction methods to remove the sequence errors or reduce the rate of sequence errors. And again, that improves the reliability of the process. So we've been doing gene synthesis commercially for about 3 years now, and we've seen a change in the way people are using gene synthesis. You know, 3 years ago, people mainly used it on where they didn't have another option, on hard-to-clone genes, hard-to-clone cDNAs, codon optimization, and design proteins. But today we're seeing some companies that outsource some or all of their cloning, and we're starting to see more use on large genes, things that are over 2 kb, particularly those that are hard to PCR. Oh, sorry. We're also starting to see more use of larger constructs, orders in the 10 to 25 kb range, and people building vectors. And today, this year, we're also beta testing a technology that's related to the previous talk, synthetic gene libraries. And the concept for synthetic gene libraries is very simple. If you can build a single fragment by adding in diversity at each of those addition steps, it's possible to build a library that's highly complex. Okay.
It's when you,
okay.
when you're close to the
Yeah.
thing, you're accidentally hitting it.
And you can appreciate, given what I said before, that this could be done either with oligonucleotide. So the little colored blocks in here can be codons or oligonucleotide duplexes or large clone fragments. So it's possible, given a single technology, to build libraries of very high complexity. And actually, that's one of the things that I wanted to talk to people about here today, is that we're interested in collaborations for testing and developing this technology. Our business model for this is just like our business model for gene synthesis. It is a fee for service. So we're interested in providing this as this diversity generation technology as a service, and are looking for collaborators who have interesting projects. So synthetic gene libraries have a number of advantages over the other methods of building diversity, and they've been really described well by researchers at Maxygen and other places. And the primary advantage of synthetic gene libraries is that the recombination is very fine. You can get recombination between adjacent amino acids. The advantages of the synthetic gene library approach that we're using is that because we have an error correction in the process, a larger fraction of the molecules are free of errors and therefore active. So those are the applications that people are using today. We're now planning projects, and we have quotes out on projects of up to 200 kb, and those are things in like bacterial operons, viral genomes, and large mammalian genes. And one of the other points, so we're now moving from reality to fantasy. One of the other points that I wanted to say is that bacterial genomes are within the range of current technology, I believe. I think that the synthesis capacity and the synthesis technology we have today is up to building genomes. We've recently bid on a 7 megabase project, and we could scale to building a megabase of synthetic DNA a month very quickly. And we're exploring ideas for genome engineering, and I just wanted to share with you one of those ideas. And it's very closely related to things that a number of other speakers have mentioned. And the idea is a reduced codon set genome for E. coli. And so what we are considering is synthesizing the E. coli genome without some of the redundant codons. And then you'd be able to use those spare codons for incorporating unnatural amino acids. And I think that this would enable a lot of different libraries to approach building, or laboratories to approach building a range of new materials, and it would provide a really economical way of producing these novel proteins. And it builds on a huge body of research from Peter Schultz's lab and others about the incorporation of unnatural amino acids into proteins, but I think would provide a relatively simple way of doing that. The other advantage of this type of project is that I think that most genome scale projects now are probably beyond our current ability to design the DNA sequences. I think we could build them, but designing a sequence that was going to work right off the bat with very many changes is at best difficult. I have a little anecdotal evidence on that, and that is that of the thousands of genes that we've done, a small percentage of those are what I would call engineered genes, where someone's taken and completely redesigned a plasmid or designed some of the parts that we built for Drew Endy's class fall into that category as well. And our observation is that with these engineered fragments, the percentage of those that are hard to clone, that cause cloning difficulties, is much higher than with native sequences. So although only a few % of our total orders have been for engineered sequences, maybe half of the things that we had trouble cloning were with those engineered sequences. Drew's smiling. And this isn't, we're not asking for function here. All we're asking is that the piece of DNA will survive and not kill E. coli or the plasmid that it's in. So I think that's just a cautionary tale on trying to design large, complex systems today. But I think recoding E. coli is a plausible first step towards genome engineering. It involves the replacement of the entire genome, but the design issues are a lot simpler. And I think it would provide a really valuable tool for protein engineering. So the current methods for introducing unnatural amino acids were developed by Peter Schultz's group and others, and they've really demonstrated the value and feasibility of adding novel amino acids to proteins. And they really involve two key components, altered tRNAs, and is one component, and then engineered or evolved tRNA synthetases. And both of these have the problem that they may reduce the fitness of and productivity of E. coli by altering the translation of the native proteins. And the idea with the reduced codon set genome is that you could provide a whole section of the genetic code that would be free from this kind of interference with the background, with the chassis. And so these are two examples of the kinds of altered tRNAs that are used. The first is a tyrosine, what's called a suppressor tRNA, where the anticodon is mutated to match a stop codon. And for some percentage of the time, it will insert a tyrosine rather than stopping the protein at that point. And the other is a natural frame shift suppressor that has a 4-base codon. And both of those techniques have been used to incorporate unnatural amino acids. But you can also see that in both cases, they would have many opportunities through the rest of the genome to alter the native proteins. And a number of different groups have described methods for evolving tRNA synthetases that are able to accept unnatural amino acids. So native tRNA synthetases are pretty good at avoiding charging tRNAs with the wrong amino acid. So there's a requirement to engineer the synthetase to specifically accept a different amino acid. And these methods have been used to incorporate many different unnatural amino acids into proteins and in some very interesting ways. So just in summary, I think the advantage of doing this by altering the genetic code is that it has the potential to improve the viability and productivity of the organism, and then thus enable industrial scale production of unnatural proteins. Some of the applications for the unnatural proteins will require very low cost production methods to be really practical. And it also could allow the use of a number of different unnatural amino acids in a single protein, and to allow those to be substituted at many positions in a protein, things that are hard to do with the suppressor tRNA approach. And I think perhaps in the long run, just as interesting, it may allow you to build highly defined polymers using the protein synthesis machinery that don't contain any natural amino acids, and branching out into really different areas. So the idea is very simple. Alanine's coded for by 4 triplets. We'd go through the E. coli genome, and in every case there's a GCA. Replace that with a GCT and a GCC with a GCG. And you'll note that I'm not proposing just getting rid of the rare codons. I think that there may be some biological use for the codon frequency. So the idea would be to try to be as conservative as possible. And this is just an example of a fantasy codon table where 12 codons are freed up in a relatively conservative way. And that requires about 110,000 changes over the 3.7 to 4 million base pair E. coli genome, depending on whether you go with the native one or Fred's shortened genome. And so we would start with the native E. coli, replace the genome with one that only uses 49 codons instead of the 61, free up those 12 codons, and then anyone could use these selective techniques to engineer tRNA synthetases, feed the bacteria the amino acid analogs, and then produce recombinant proteins with these changes. So I just wanted to briefly touch on our thinking about how to build a bacterial genome. As I said, I think that the design issues are probably the hardest parts at this point. We would propose to synthesize modules, probably something in the size of about 200 kb, and then sequentially substitute these fragments into an existing genome. And one other issue would be with the quality control, and Fred touched on the use of gene chips for this. I think with the kind of changes we're proposing, using array hybridization would be a good way to track the whole process. There'll be single nucleotide polymorphisms spread across the entire genome that would let you at very high resolution track the substitution of the altered genome into the native genome. Another, I think, important component of a project like this would be to check the functionality of each of these pieces as you go along. And one method of doing that could also be based on the use of gene chips, and it's similar to work that's been done in Ron Davis's lab. And the basic idea is to take a section of the genome, substitute, say, a 40 kb fragment of the altered genome, and mix two isogenic strains, one of which has the completely native genome, and the second of which has the segment substituted in, and then put it together a 50-50 mix of those two genomes, grow them up under the conditions that you're interested in, like fermentation, and then measure the frequency of the SNPs in the N population. And that gives you an extremely sensitive measure of the selective, of the competitive ability of that segment of the genome. And I think it may be necessary to have sensitive measures of function when you're trying to re-engineer a genome, because the last thing you'd want to do is get 3/4 of the way through and find out that you'd accumulated enough small deficits in function so that you couldn't go any further. So in summary, we built a large number of genes with the technology that's available today, including many fragments in the 10 to 20 kb size range, and we're starting to think about back-sized projects. And I think that the gene synthesis technology it's really improving rapidly, but today it's adequate for tackling genome-sized projects, and that much of the complexity is in the design. And I just wanted to mention one other thing, which we're calling the Big DNA Contest. And so what we want to do as a company is to stimulate people to start thinking about using large synthetic DNA fragments and to test a new technology that we've got an SBIR grant for assembling those large fragments. And so what we're looking for is ideas for a 30 or 40 kb fragment that's scientifically interesting, of use to somebody outside your lab, to a range of other groups, and requires or benefits from synthesis. And basically we're looking for a 2-page essay on why this is a cool thing to do, and the prize is we'll build it for you. So you get $100,000 in synthesis to build it, and we'll give you the clone for the person who has the most aesthetically pleasing 40 kb suggestion. Okay, questions? Yeah, Roger?
As you so rightly indicated, it's the rare piece of DNA on a plasmid that kills an E. coli. After all, why should it? You know, it's much more that the piece of DNA keeps the plasmid replicating in some way. Now, given that the ability to clone and propagate any old piece of DNA, one that's not been through the fires of evolution, is of clear interest to this community. It strikes me that Fred Blattner, your report on that poor German guy who had that sequence that was just this transposon magnet, it raises the issue that some pieces of DNA may not be stable in clones because they're magnets for something that's indigenous to the E. coli. Those you want to get rid of by using a different E. coli. There's other kinds of instabilities, inverted repeats and so on. I think it might be part of the agenda here over the next year or two for people to parse out more precisely the kinds of things that make a sequence unclonable than has been really done thoroughly up to this point.
Other questions? We have one over there.
Yeah, given that it's pretty hard to set up a recombinant DNA lab in a cave, one could imagine certain groups of evildoers who might be really keen to outsource their cloning. Are there any steps in place to make sure that Blue Heron doesn't unwittingly provide such people with the next bioweapon?
That's a very good question. It's actually one that we've been thinking about since the founding of the company, and actually way before 9/11, although it's certainly come to the fore since then, the anthrax scare after that. Basically, we do two things when we see a sequence. One is to analyze it for how we're going to build it, and the second is to compare the sequence to a list of pathogens to make sure that we're not inadvertently building a pathogen. So I think that today, I think it's relatively unlikely. I think there's a lot of easier ways to get your hands on a pathogen than have us synthesize it. And you could certainly imagine ways in which you could design around this scheme that we're using today. So I think that over the next few years, what we'd like to do is work together with other groups to try to develop methods for identifying really novel pathogens in our order stream. That may or may not ever happen, but it would be nice to have the capability.
So are we correct in assuming that it's around $3,300 per KB?
So the price right now is $250 if you're an academic, $275 if you're in a company.
Per
base.
Per base. Then the second question is
I was thinking 40.
is interspersing natural sequences with the synthetic sequences. Do you do that also?
Yeah, we call that, we do that as a sort of beta test now for some of our customers. We call it partials because one of the things that people often have is a partially finished cDNA where they've got 9/10 of the clone and can't get the last piece. But yes, the technology, especially the solid phase, the second stage solid phase assembly is really amenable to putting together mixtures.
Sure.
Synthetic modules. I think there's lots of interesting potential applications that we haven't really thought through or got people thinking about.
Okay.
Okay, we have a question back in the corner. George Church.
John, you mentioned error correction a couple of times. I wasn't clear on what the method of error correction was. Was that just sequencing intermediates, or was there something else that you had in mind?
Something else. At least two of our competitors are here today, so.
Okay, other questions? Okay, if not, let's thank our speaker again. And we'll recess for a short break.
Number 5. This should be 5, Phil. This is line 4. Anything? 412. Mic 4.
Sure.
You want to go ahead and kill the mics and we'll scoot the table.
discussion around intellectual property. The time here until 5 is meant to be an open discussion amongst all of us. And so just to get started, the framework for the discussion to start off with, at least, will be as follows. I'd like to spend a little bit of time talking about applications, classes of applications that we might be interested in going after, and use that as an example to begin to better specify the sorts of things that we're going to care about and thus might want to protect in the context of intellectual property. From that substrate, we'll try and move into a discussion of ownership of biological information or the physical artifacts themselves, and what the features of an intellectual property framework might look like if we could start from scratch and specify something that we'd like to have. We won't be able to address the problem of how you would actually do that and whether it would be possible, but we'll get to an end point, hopefully, where we can bring up some of the tools that we have at our disposal and try and consider whether it makes any sense at all. We're going to stop about 15 minutes before the hour and turn things over to Roger to pre-position some materials for tomorrow's risk discussion, just to get the thinking going there, so it's not all coming at you at us at the last minute. So I'll give one example of an application that I'm personally interested in, and then I think we should just try and open it up and see what else folks might want to make. So this idea actually comes from a discussion Roger and I had a couple years ago, I guess. Is the idea of being able to implement counters inside cells. And so, for example, if you could take a counter, a 4-bit counter, and put that in a worm and couple that to cell division during worm development, you would be able to track events as the worm went from a single cell to 959 cell animal, things that were coupled to cell division, and that would be cool. And if you had a 5-bit counter, you could put that in yeast, and you could study onset of senescence in yeast instead of staining with calcifluor on the bud scars. You could just count and collect cells and do microarrays and maybe reprogram gene expression as a function of age of the cell. And if you had an 8-bit counter and you could count up to 256, maybe that would be cool for studying aging in humans or onset of cancer or who knows what. So these are examples of what other engineers would consider to be modest information processing devices. But they're of a scale that are more complex than the artifacts that we're currently able to produce. I don't know this to be true, but it feels likely to be the case that were I to try and make something like this, I might want to make good use of dozens of synthetic transcription factors from Sangamo or Carlos's lab. I might want to make use of dozens of engineered proteins from Wendell's group, and so on and so forth. And so I'm imagining we're heading to this future where we're going to want to be able to make use of parts in combination. And it feels to me like the current situation is that individual classes of parts are being binned up. And so that's an example of a system that I think is cool and would be useful. And I'm not sure how I'm going to get to be able to make it. But before we get to that question, do people like or dislike the counters, or do people have other systems of modest complexity that they'd like to go after or think, are planning to do? So sorry to put everybody on the spot. It's really meant to be a discussion. Yeah?
Sure.
There's a mic right there.
I think one of the IP issues is what's patentable and protectable. One kind of patent that has been traditionally very strong is the material patent, meaning the actual physical material chemical composition. For many designs that I think we're considering now in synthetic biology, the material composition is almost irrelevant, that you can do the same function with a multitude of different particular materials. And so I don't know where IP and patent law protection is going to treat the pure, ephemeral, non-material design issues.
Do we want to deal with that now, or do you want to?
Let's table that. Yeah. I think so. Just to restate that, if you needed a switch in your system, there might be many ways in which you could implement the switch, and so it's not absolutely relevant which particular sequence information is encoding the switch. So let's come back to that issue, information versus function.
For what it's worth, nature's switch is a telomerase, a counter is a telomerase.
Fair enough. So you know.
Well, is it your intention to introduce an artifact, Drew, just to kind of lay out the sets of issues, IP issues that might be associated?
So the counter is one initial artifact, but
Artifact?
something
thing, human-made thing.
some physical thing that I as an engineer construct.
Indeed.
So, but are there other, you know, what systems of modest complexity could people foresee wanting to build?
Okay, so to give you an example, a set of things that will express your favorite protein at a specified molar level.
But I think it's also possible to sometimes cover a whole set of applications by just claiming all of the combinations of the parts, and so you don't actually have to know all the interesting bits, all the interesting combinations specifically. But if you have a description of a set of promoters, a set of ribosome binding sites, a set of proteins, and a set of receptors, for example, then you can essentially, and you have specific examples of this combination being useful, this combination being useful, this combination being useful, you are likely to be able to get a claim that covers all the combinations between this set and this set and this set. So it's a claim that has one promoter out of the set of these, one terminator out of the set of these, one gene out of the set of these, and any which way of hooking them up together. That's the kind, in industry essentially, that's why you try to get a broad claim coverage essentially by going as combinatorial as you can. And of course patent office is aware of this and they try to cut it back, but it's this is a way to essentially get broad claim coverage. So by describing the sets of elements in different classes and then claiming the combinatorials.
Okay, maybe one way to ask this question would be, let's assume this artifact, this counter or whatever it is, then what would one want to have associated with that artifact in terms of an intellectual property regime or a set of expectations about how people who assembled that artifact from sub-assemblies treated those sub-assemblies, etc. I guess I'm asking this as a question, is a counter example good enough to go on with?
I was going to answer Drew's first question, but I think the counterexample is pretty good for the reasons that the other person from the audience mentioned, that it could be implemented in using different materials. So it's a very good example. I was going to give another example of a smart virus that could execute a series of conditional statements within a cell, depending upon what it was designed to do. For example, to identify and remove HIV from an infected cell. So something of that order. That would be a medium-scale system, I believe.
Okay, and that would be perhaps a bit more ambitious, an engineering project. But I mean, that's okay.
But that's what they're after, then.
Yeah.
Or is it,
Okay, so well, why don't we?
Yeah.
So one of the questions that gets raised when you think about the process of engineering new biological systems is what is it that you're concerned about? What is it that you'd like to make available or protect, right? And so you can hear three things that you can use to break it down. One would be the physical material itself. The other would be the information specifying the physical material. And the third would be technologies that are useful for either discovering the information or assembling or debugging or manipulating the systems. So one question to pose, right? I mean, some of these questions, the answers seem obvious. Are you concerned about the physical information, the physical material, or the information specifying the material? At present and in the immediate future, it feels likely that the physical material matters because we still have -80 freezers. But if John and others develop synthesis as a technology still further, the information becomes limiting, right? That's, I think, John, your comment about design being hard. Knowing what to make isn't trivial. One of the questions that comes up, though, is what's relevant here, right? Are you really concerned about maintaining access to the information or protecting the information? Or are you more concerned about controlling the technologies that are used to construct or use the information? So I don't know if you want to come in on that, Roger? Or?
Well, maybe it's time, it might be time to break it out a little bit. So in the regime of patents, you have, you know, in biology traditionally, you know, this starts with the chemical companies, the pharmaceutical companies, and so now you have particular configurations of atoms. Then you have classes of patents that apply to things which somebody from an engineering background might not know about or even think was a good idea at all. You can patent the idea of nailing a protein, a eukaryotic protein to DNA using a prokaryotic DNA binding domain. You could, I don't know if you did, Wendell, but you could certainly apply for a patent on the idea of a scaffold protein that sent a different protein kinase into the nucleus in response to a signal that should have activated yet another protein kinase. So you can patent broad concepts. You can patent methods, such as pulling magnetic beads to the side of a tube using a magnet. Okay, and all of that gets, so you get configurations of atoms, you get ways of going about doing things, you get broad descriptions of those things, and then maybe, Pim, you get combinations of things where you see from these few examples that we've combined that we could do any of this.
Actually, there was actually a recent ruling in 2004, there was the BMS case, the COX-2 inhibitor case, where it was this rule that essentially there needs to be substantial description of the molecular composition of the agent that does something. So in the context of therapeutics, at least, if you describe how to inhibit a reaction with a compound sufficiently potent to inhibit the reaction, that's not good enough. You have to, so you list, typically there's a lot of patents out there that list the interaction you want to inhibit, but they have just a very general description of the kinds of molecules that would do this, small molecules, proteins and others, but they don't actually describe exactly which protein does that. And so these claims are not held to be valid any longer. And so this is, well, the claims still exist. People are now starting to essentially assume that these are not valid. And so in order to get claims that where you describe the component that does something, you'll have to describe in detail there's a description requirement for the protein that does that. And so there's a specific exception to the description requirement for antibodies. So antibodies, you can claim any antibody against the target. And that's why we're not working on antibodies for therapeutics. We're using a non-antibody scaffold so that you don't have this limitation. But so you would have to, and this is more clearly developed in therapeutics, but expect logic is similar here, that you'd have to have substantial detail on the composition of the molecule that is going to perform the action. And you will not, and you may be able to get a claim that's much broader than what you've shown, but not all small molecules, all proteins, and all other agents.
Right. What I'm taking, the way I'd laid this out is that you're getting a sense now, a scattershot sense in a few examples of the regime that now exists in the world of biology, and you may not wish to port all aspects of this marvelous scheme to this new world you're saying you want to build.
Yeah, I was going to say that I was a bit confused by where the conversation was going, because it sounds like you're giving us instructions in how patent law works, but it seems like what we would want to discuss is how to make the whole system mimic the open source Linux ideology as much as possible, and how to basically prevent the situation that has arisen, for example, with phage display, where basically aggressive patent lawyers have castrated what is an ideal technology for doing things. And so, I don't know.
Hear, hear.
Mm-hmm. Let me propose at this point what I might call the recording artist model. All right. So the the problem here is the balance between some return for your artistic creation. Otherwise, there's no economic incentive to do anything. And the obnoxious situation where patent lawyers and patents tie things up unreasonably and beyond all belief and extract or extort the maximum possible price. So the recording artists, they make a song, and radios play it, and the radio stations pay a set, very small, preset price each time they play the song. And so I think the features that are desirable about this is it's related mostly to the exact physical object, although you can also have some rights on the lyrics. So if you cover it by another artist and so on and so on, you get some variation, and both the original author and the recording musician get some things. So you can modify previous rights. And that there's some arrangement where the technology is freely and immediately available at what is ultimately a very low price, perhaps tied to a small fraction of, say, the ultimate sales price if there ever is a sale.
Right, right. So that's called the BMI or ASCAP model from the recording industry. And that does, among the features of that, you cannot keep anybody else from using your song in their movie. You cannot prohibit them from doing it. You know, one disadvantage, you have to keep, now you have to set up a chain of provenance. Who did what, who wrote what lyrics, and you have an elaborate cost recovery scheme. But it's not a, you know, it is a model. It worked well in 1985. That was working well.
Hey, Roger, but you can keep somebody from using your song in a movie. It's copyright.
There's an obligatory exception for I look at music shuffling, and there's an obligate exception for obligate licensing requirement for music.
Right.
And that doesn't apply to any of the other copyright categories. So I looked at, it's an obligate licensing requirement for music.
Right.
But it doesn't apply to other categories like art. And,
so everybody had to pitch into this consortium, but then everybody gets something else.
So there's another model that might be interesting to take a look at, which is the semiconductor manufacturing model, where basically every company has a pile of patents, and they're patents of their very own. They're not they're not licensed in any direct way, but everybody's cross-licensed to everybody else. So if you go and you make a chip at Intel has agreements with all of the other manufacturers, and there really are not any patent issues. I mean, the manufacturers settle between themselves. Basically, they put their pile of patents up against the other guy's pile of patents, and some money changes hands. But the end users don't really see that at all. So you could imagine if we transition into a situation where the DNA fabrication becomes centralized, that a similar sort of cross-licensing approach might be a way to handle some of these problems.
Yeah, so that requires that it be big, that each of the 7 people who plays in that space be big, and works against, and I'm going to be agnostic on this, the development of a hacker-type culture insofar as you want that to develop.
So I want to come back then to the proposal or comment that was raised earlier, and if you were to sort of step back and sketch out a wish list for what a IP framework might look like in future biology that is inspired by, may or may not be the same as an open framework. Here are some things that it could account for. So you might want to have unfettered access to the information specifying the components. So you could simply read and learn about stuff and know what was possible and what was out there. You might like to have unfettered ability to use parts in combination. So I could take one person's zinc finger and another person's engineered protein kinase and make good use of them. I might want to maintain a chain of authorship, and that could be distinct from responsibility. So if you're worried about accidental or intentional misuse, you might want to know where things are coming from and who did what. I'm likely to want to be able to promote the development and use of higher level descriptions, what a biology, an annotation of sequence. All I mean to say is, primary sequence information is often not human readable without a tremendous amount of additional work. And I might also want to have triggers that promote transparency, so that if the information specifying a system or the physical system itself was released into the environment, that all knowledge of that system and how it worked would also be made available. So this would be analogous to, for instance, how the free software licenses work. When the binary is released, the source code must be released as well. So I don't know that this is right or not. It feels, if I were to try and implement something like this, a starting point. And so I put it up in response to the comments from before, but I'd be happy to try and modify this. I have no idea how to implement this and whether it should be implemented at this point.
Yeah. Okay, testing. Okay. So, the extreme case. What happens if you have a company, they haven't patented any of their, or they're seeking? Well, what type of patent would protect a company which designed their biological sensor or medical diagnostic thing, and they're making money, and this other company then just buys their product? They could reverse engineer it pretty quickly, or even just culture their own organism and resell it. And what sort of patent would protect that from happening?
Most sorts of issued patents governing biotechnology do protect against that.
So what happens if they reverse engineer it, change a few things?
Doesn't count. Still patented. Still protected.
So at what point is the variant no longer a variant, and a variant is now a new design? And
it's established by patent lawyers and interacting with the patent office, and the bounds are rather wide. So it is possible, and there's an issued patent covering bringing proteins to DNA by a prokaryotic repressor.
So
you can't reverse engineer it. You can't mutate that prokaryotic repressor and not violate that patent within very broad limits.
Okay.
And many of those overly broad patents actually never really become valuable. There are overly broad patents around, and the Kaufman patent, for example. The Kaufman patent.
No, but explain.
And so it's...
what is that patent?
Well, there's a very broad... The Kaufman patent is any random mutagenesis of proteins, random proteins. There's a number of other they essentially they're useful as negotiation pieces, but they're generally not licensed. Nobody licenses them.
But that's going against the whole hacker culture, right? You have an unfettered broad patent protecting a lot of things, and of course you're not allowing anyone to use it.
Right.
So that's bad, right?
So I just wanted to maybe expand the thoughts about the IP framework. If you go back to the original notion of what IP is and why IP is guaranteed by the Constitution, it's to enable continual innovation. It's a promise that you will be able to keep doing what you're doing and in fact do better for some period of time. And one of the issues that's associated with that, I believe, although not listed up there, is how fast we want to go, or how slow we want to go, how slow we want to enable, or how fast we want to enable innovation to go forward. And if we can control that at all. I just want to throw that out there and add it to the list.
Oh, okay. So, so Drew, I wanted to expand a little bit on Rob's point there. The reason Thomas Jefferson created patents as eventually was implemented in the United States was to actually create an open source system of a kind. So the point of patents is that you would know exactly how the technology was implemented in order that other people could use that to make new and better implementations. But with the commercial rights being assigned for a limited amount of time so that you actually could get some commercial benefit. So that actually is very reasonable, and because the alternative is to use trade secrets as a way to protect your intellectual property. And if patent law does not give sufficient protection to any particular invention, then the maker may decide not to tell you how it works and to use all methods possible to obscure the way it does work and use trade secret protection, which then obscures the information and makes it unavailable for others to make progress. Okay, so that was just a broad issue. Now to get back to your original question, I think when you talk about access to use parts, I think you should divide the issue into two parts. Access to actually play with the parts, or access to play with the parts and then commercialize what you want to do with them later. So, to play with the parts, I think it would be a very natural extension, if it's not already true, that if you use the parts for non-profit purposes, that that you should be allowed to do so. And I'm not sure that patent law, I'm not sure exactly, we should ask the lawyer here what the exact issues are, because I do remember people being sued for using
correct
Taq polymerase at one point. So, but I think that that small change to patent law to allow nonprofit use of the material would be reasonable if it's not already the case, as I don't know. And second, the commercial aspects should be separable in this conversation. That's just clear to me. And then, of course, that has to be worked out among the people, among those who are interested. Anyway.
So I just want to acknowledge what I'm now hearing as two additional points or modifications to this initial naive specification. One is something having to do with the rate of development or application of the technology, and the second is access to all of society's resources, basically. You know, profit, not-for-profit, whatever.
Yeah, to get a little technical on what you said, Andy, all of us kind of thought there was a so-called research exemption, which allows you to use any technique. And the courts ruled quite decisively, I think in 2003, maybe even 2002. There's now a federal appellate court ruling that there is no such thing. That Duke was judged to be a money-grubbing institution in business, and the business is writing papers and getting grants, and you guys have to pay royalties.
Yeah,
I believe that patent law explicitly does not allow you to do anything other than study the process described by the patent. It is not, you cannot use the patent for any useful purpose, including your own research.
There's no research exemption, yeah. But that could be made explicit in some.
And some countries have this. So Denmark, for example, has a research exemption. Sure. And that's why we have a subsidiary there. Yeah.
Yeah. So I want to get to Paul, but Rob, did I get that wrong or something?
That's fine. I also just want to throw out that the notion that patents are property is not an old notion.
No,
so one of the things that Lessa keeps pointing out is that in the sort of space of the internet and associated technologies, that what he just over and over calls patents, the thought that patents are property is absurd. And this is not something that was originally intended to be the case, but it's developed to be that way. We could decide as a matter of policy to do it differently in the future, although that might be problematic economically.
Well, what was it? The patents were never property. They were a license.
A patent.
Just in that line, a few broad points to keep in mind. Trade secrets, patents, but the third alternative is putting things into public, which if you're worried about too much private control over what you're doing, you can publish them and put them into the public domain yourself. That means you can't make money on them either, but it means they can't be taken out of the public domain, at least not easily. So that's step one. So that's worth considering as a collectivity or as a set of individuals, whether you might not want to take your registry and make it public and publish it as quickly as possible so that somebody else can't patent it too quickly. So that's step one. Step two, we're already in a global situation in which U.S. patent law is not sovereign and autonomous and dominant. So the European Union has clearly some different ideas about what the scope and limits of patent law is, and the recent BRCA1 decision where this goes with Pim's point, where since Myriad made some minor mistakes, they invalidated the patent. This is in the direction that you really have to, the bar is getting a little higher. So European Union's a second player in this, and then the third player, which is still completely unknown, except is where China and India and other places are going. And that issue turns on, as we've seen in the AIDS epidemic, whether or not there are any general sense that there is a global commons or a global set of of goods which can limit corporate rights. And in this case, those battles have actually basically been won. So and now that the intellectual infrastructure to do world-class molecular biology and other sciences is now being distributed around the world and is not controlled by the US and Europe anymore, I think we're entering into really a quite different domain in which 18th century patent questions, strange enough, may be somewhat more relevant because there were Jefferson and others in the 18th century did have a sense of the general good in a way that's been rather undercut by industrial capitalism over the last 2 centuries. So I think you need very specific thoughts about what to do in the next few years about with what you're doing, and you need a longer term, I mean 10 or 20 year perspective about what this new global situation is going to do to the kinds of inventions that you're making.
So I think one possible solution to a lot of these problems, to the potential limitation of research by companies. Some of the conversation has been towards the idea that there are consortia that get together and cross-license and allow each other easy terms, but you never can exclude the possibility that some evildoer corporation will patent something and decide not to be part of a friendly system. They will patent some essential aspect of what we're trying to do and basically hold everybody hostage. And I think one way to address that is to have something like the parts collection, which is going to be central to what we're all doing, be, for example, patented by MIT, and it would simply be announced that this is freely available for research use, and for commercial use you have to talk to MIT. And that way, anybody who tries to further patent things based on the parts could be prevented from doing so by MIT. And that way you could ensure...
But I think the idea that it would be,
I think the goal is to ensure, first of all, that research use is never inhibited.
That's right. But the assumption is that we have to rely on the continued benevolence of MIT.
Yeah, the world is much bigger than MIT, right? And there's not a chance that MIT, deploying all of its resources, would be able to develop the set of components that we'd like to have. So that doesn't seem...
So what do you propose happens to the parts? They're just in the public domain?
Well, so I'm doing a bad job of trying to decouple the problem into one. So I want to get Fred the mic real quick. But decouple the problem of what would be the features of a framework by which we would make use and share biological information and materials. And then the second part of the problem is, what are the tools we have to implement that framework, right? So probably the more important part of the conversation for us now and throughout the meeting is to have discussions about what is the specification of the framework that we'd like to try and implement. And then, I'm certainly not qualified to figure out if it's implementable and the best way to do it. So we can think about that, but work towards that later. So.
Well, I was trying to warn you that MIT is also a corporation, and they actually probably own what you've got already, because if you had federal funding in it, they would. If you don't have federal funding, then it's complex. But the Bayh-Dole law basically says that if you've developed it with federal funding, you're obligated to put it in the hands of your institution.
No, can you pass the microphone over one?
Let's hear what you say.
Well, I can just say that that I have an agreement with MIT. I'm a faculty member at MIT. I've been 31 years. I have an agreement with MIT that all of the work that I do becomes GPL. That goes to the GNU General Public License, and that has the feature that it then becomes free to anybody to use, so long as it propagates the GPL work. Now, whether that would work for patents, whether that would work for biological materials, I don't know, but I'm just saying that, an example.
You're talking about software then? Do you want to talk about that?
Yes, but also I also have...
And I want to limit the degree to which we're just arguing with each other and try to get the principle that whether I've got it exactly right or just approximately right, there is a Bayh-Dole law, and as far as patents are concerned, and they continue to extend this, because it really doesn't say it's limited to patent, that the university is the agent that is considered to be in a position to, and look, this hasn't been tested. What if they go? Can they go and confiscate your notebooks and patent your work? I don't know. But I wanted to get my suggestion out, which is that if you want to set up a new framework, you probably have to get a new organization that would be authorized to collect the IP, other than MIT, and MIT might fight you on it. They may actually want these inventions. I'll tell you, I could see plenty of applications for the counter in someone who, let's say, is selling a biological bug, and they want to charge you by the amount that you replicated. You could
subscription
use
model
it for a very commercial purpose, a very simple idea. It could be used in a very benign, I would say, sort of like a taxi meter.
So let's go back there, and then new bar.
Way up here?
No, right by the stand. No, by the Heather, by the stand.
Yeah, I would like to say I think the GPL is an excellent sort of model. I mean, it's probably got a lot of holes in it as far as molecules go, and a lot of things that are software specific. But in general, I think it's a very good model for the kind of thing you'd like to have to govern the way that the different parts in synthetic biology are used. And one thing that shouldn't be overlooked is that the open source doesn't necessarily mean it's free. There are a lot of open source programs that generate money. I mean, a lot of file servers and you know, web servers and things like that are open source, but people still pay for implementations of them or to have them, you know, ported to a particular platform or whatever. And the only thing the GPL constrains you to do, you can charge for your software, but you just have to propagate the GPL, as the previous person pointed out, and that also ensures that your points, Drew, about chain of authorship and transparency and so on. The GPL actually forces you to acknowledge who produced different parts of it, but it doesn't mean you can't pay for them. And people do pay for things that are, you know, that could be free, because there's an awful lot of time required in figuring out how something works and implementing it and so forth. So, you know, open source wouldn't necessarily mean uncommercial.
New bar?
Yeah, just wanted to add, I don't know if you can hear this. I just want to add one thing to this, and maybe there are experts who know the answer to this, but it strikes me that the more you're engineering with natural parts and you're trying to do things, the more there may be an argument to be made that this is precedented in nature, and the composition of matter claims that you may want to get may actually not be available. So the EST patents, certainly people raise those kinds of things, but I think it's important to try to think that through. If you're doing an unobvious combination of parts, but it has already been done, and you can find evidence for it, I don't know how you can actually get the most fundamental patent. And I think once you kind of eliminate that possibility, all the other method of use patents and all that will be much less of a block than people perhaps are concerned. So it would be an interesting question from my point of view, if there are experts, patent law experts, as to whether the reliance on these natural parts or kind of equivalents of those, slight variations of those, the patent law is pretty clear, don't count, whether that you can make an argument of their pre-existence.
Eric?
Yeah, I'm not sure I understand the scope of the problem that we're discussing. So let me put out one example, and then in comparison, maybe you can help me. If I create a comprehensive knockout library of your favorite organism, and then deposit that library in the ATCC, the American Type Culture Collection, right? Then that library, I'm putting it there to make it available to the research community, and to give them to put the burden of distribution on them. In what sense couldn't you do that for these biological parts, devices, and systems? Deposit them somewhere, give somebody else the job of distributing them, and they, you know, that at least at cost.
Yeah, I think that's easy to imagine. There are lots of other issues, though. For instance, I myself am never going to construct or discover or invent all of the components that I might like to make use of. And so how do I implement a situation where, and I presume, that we all have.
But you just need a magnet for the part. They wouldn't all come from you. I mean, Barbara Bachman back in the days, in the 60s and 70s, collected the E. coli mutants for the community, and there are such curators now, I believe, for most of the model organisms. Why wouldn't that work for the parts, devices, and systems?
It's clear to me that this discussion is not going to be able to do anything except illuminate issues, touch on them really. But one of the differences, Eric, is that that I see is that when you're bolting together software out of pre-existing pieces of software that's protected under various open source licenses, and the bedrock, this one hasn't been raised yet, so I'm just going to try to throw out another one. The bedrock on which the GPL and its derivatives rest is copyright. I assert copyright in the code I wrote, and I am giving you a license to use my copyrighted material if you adhere to these conditions. So the equivalent of copyright is a way to copyright words and lines of code. The equivalent of copyright in the corporeal world is patent. And copyright costs you nothing to type a C. Patent costs you $50,000, something like that. So just to assemble an open source thing that's, closely based on what's done in software is going to cost. It's going to cost so much.
That's why MIT had their whole operation set up the way they did.
Okay, I want to take Carl and John, and then we're going to table this.
So I had one question, if you follow up on the point, Roger, that you just made about copyright and patent. Do the combinatorial aspects of creating complex structures actually differ in these two cases? So if you follow through copyright, you combine A and B to get C, and basically some combined system infringes if it plagiarizes any piece. If you combine,
right, that's sampling in music, say, as opposed to bolting together copyrighted pieces of code protected under the GPL.
Yeah, so say you bolt together those pieces of code and make a new thing and claim that that's somehow yours. You can't do that because it's clear where the pieces came from. It is not clear to me if you bolt together publicly available things and make a new physical object, that actually may be patentable because it's a new entity. So the logic of combining pieces may be fundamentally different in these two cases.
Okay, John.
I just wanted to mention two things that relate to this. One is the material transfer agreements for physical objects can be much like the copyright on music. Material transfer agreements can last forever, but they only follow the chain of the physical object. The other is, I don't see any fundamental problem with a coexistence of an open source materials where you publish and make unpatentable segments of what you use and allow commercial entities to make inventions using maybe those segments and their own inventions or their own inventions alone. It seems similar to what's going on in the software world and a really feasible model. You just have to make sure that the pieces you're creating and distributing are published and clearly not patented and therefore not patentable.
So I'll take one more from Paul, and then I want to
keep in mind that the SNP consortium was put together by 9 or 10 of the biggest pharmaceutical companies around to overcome some of the over-parcelization of small patents that the biotechnology industry had created because they didn't have products, but they had patents. So you've got to think about this historically as well in terms of how value is generated in the parts of the world you're talking about and how that's changed. So capitalism is made up of multiple different entities that operate with different interests and at different times. So that needs to be thought about as well. And the SNP consortium is not the only one of its kind, where commercial interests have banded together to overcome lower level patents, and the changes in the patenting office in Washington are being pushed by big money to eliminate other kinds of patents which are standing in the way. So you need to know in some detail which commercial interests you're talking about and how global they are.
Okay, so first, I'm sorry, we're going to need to move on right now, but so thanks for participating and putting up with an unstructured discussion. The most important thing I think that's coming out of this is the specification, the beginning of a specification and future refinement of some framework, what we would like to have happen. If you have more comments on that, please let me know about that. So for the last 5 minutes before we break for the poster session, I've asked Roger to briefly set up tomorrow's conversation on risk in biology. So this may be a risky endeavor, but the hope here is to just begin to start thinking about that, and the poster session will follow soon thereafter.
Okay, thanks. Next slide. Okay, just so for 5 minutes, introduction to the situation in synthetic biology as I see it, an example activity that a reasonable person might be accused of constituting, might think constituted a risk in the colloquial sense of that term, an example framework for thinking about what I'm going to call a problem, an example tactics, and that's the really important point here, tactics that one might consider using to try to ameliorate the risk. Next slide. So let's compare and contrast with Ensilomar. Now, in the middle 1970s, the upsides of doing recombinant DNA research were perceived as very high by the establishmentarian scientists who were trying to promote it. They foresaw that in the near term that it would be possible to make large quantities of human proteins of therapeutic utility, and that there was lots and lots of money on the table. The downsides were generally perceived to be low. Most people thought they were probably low, and some of those downsides were addressable by directed short-term research and engineering projects. Can naked DNA be transmitted to mammalian cells? The experiments of the late 1970s said no. Now we think yes. E. coli, is it possible to make an E. coli that wouldn't grow outside the laboratory? Yes, that's going to be a short-term project. Next slide. Compare now. The perceived upsides are not yet well articulated. The so-called killer app is not necessarily there. The artifacts, the counters and so on, are compelling, I believe, but they're compelling in an austere way. They'd enable research. There's also some difficulty that I've had for many years distinguishing stuff that's called synthetic biology from existing engineering activities that use biology, biotechnology, the chemical industry, etc. So, and there's no strong synthetic biology specific markets yet articulated, so there's no greed and lure of lucre of the billions of dollars driving the scientists here. And I'm going to say, by comparison with the 1970s, it's easy to articulate scary scenarios, and in fact, there's no particular reason to conduct any applied research because we know that these are real. Next slide. Okay, so this is a different DNA company, John. This is from Glenn Evans. So this is a large-scale DNA setup. Next slide. So here's, I'm just going to throw this out. Okay, so SARS plus. Okay, it fits your contest, John. It's a positive strand RNA virus. It's under 30,000 base pairs. So we're going to make SARS plus. We're going to resynthesize it, and we're going to add a toxin. Now, SARS is extinct in nature, we think. Okay, and it's pretty lethal, and it's extremely contagious. Witness, it keeps leaping out of laboratories in mainland China and in Taiwan. So we're going to resynthesize. We're going to make the double-stranded DNA by full synthesis or by serial recombination in E. coli, or in yeast, or mammalian cells, or plants, or whatever. We're going to make the RNA from the DNA to get live virus from the tissue culture cells. We'll have something that's more as contagious and more lethal, and then we're going to infect the hench people, and they're going to cough on people, and then an epidemic will start, and people will die. Okay, next slide. Okay. Okay, so now what I think we ought to try for here, I'm going to propose this, and I'm going to throw out some examples, is that you know, this group, if it feels self-conscious and self-confident, should articulate a vision in which the plausible opportunities enabled by this are grand. and in which the perceived risks are mitigated by some patchwork of measures. Further, that this vision becomes a statement of your strategy to gain support that the work should go forward. And then those individual measures become become some of your tactics. So that's, I'm going to propose that's the strategic task. Articulate a vision in which the potential is grand and the risks are well managed. Next slide. Okay, now I'm going to throw out some example tactics, and people in the room may not like them at all, which is part of the point. You can't make DNA without a license. Amateur radio operators have to study for tests and qualify for licenses, so do pilots. Writers of computer code do not. Which should it be? Should 16-year-olds be able to synthesize DNA? Yes or no? I mean, this is something on which I believe you need to have a stance. Next slide. The emerging synthetic bio doctrine, which I heard about from Tom and Jerry and Drew and so on, wants to decouple design and fabrication because that's a good thing for various reasons connected with how the semiconductor industry played out. So maybe we want to promulgate social norms, making it wrong for most engineers to recouple. Maybe we want to deliberately restrict knowledge about fabrication to research scientists or some subset of engineers who are, quote, trusted, end quote. Next. I'm not advocating any of the, I'm saying that you got some tough things to think about, okay? Here's an example. Extreme criminalization. Morris releases the internet worm. He didn't intend to do any harm. He got sent us to community service, which he did not serve. And he got a job at a company he started, which he then sold to Yahoo with 3 of his confederates for tens of millions of dollars during the mid-boom, say 1997. He didn't intend to do any harm. Any bio-Morris, and of course any person or commercial entity that aided him, would already be subject to the mother of all civil liability lawsuits. In fact, I'm going to say that failure to address this point alone could have what the lawyers call a chilling effect on the development of this field. But in addition to civil liability, do we need to say, build and release something that causes harm, and whether or not you intended it, you're going to go to jail for a very long time. And if they ever let you out, it won't be like, oh, you got that hot job in the biotechnology company doing security. No, it's people will spit on you in the street, about like if you were a child molester. So the normative and criminal regime, in other words, de-glamorize outlaw hacker culture now. Normative and criminal regime, this would have to be international. And it's much like the international system of norms and laws that governs world attitudes toward slavery. The world has a common position on this. It doesn't tolerate it. Next. Back to the framework. Remember your articulated version of the possible benefits from developing this field, plus your assembled collection of normative and technical tactics to ameliorate and address the hazards. That becomes your strategy for winning support that this ought to happen.
So we'll come back to that tomorrow. The poster session starts right now, and it's just outside the auditorium. You saw it earlier. And I believe dinner is at 6:30? Yeah.
Should be a stand-up comic.